Good morning, everyone. First thing first, I want to tell you that I think our class is not gonna do photosynthesis. So we're gonna do one last chapter because of all these online thing, and hopefully that will help you guys out a little bit. Um, First thing first. Okay, today we're going to talk about a new chapter. So hopefully you understand meiosis or meiosis. That will help you to understand genetics. Okay, if you don't understand meiosis, you will have a hard time with genetics too. So you heard of the name Mendel. Hopefully he is the father of genetics. And at that time, people are wondering, they all see that kids look similar to their parents, but they don't know um, how. So there are two hypotheses at that time. One is blending inheritance, which basically means that the characteristic of both parents kind of all blend together and become the offspring, right? The other one is inheritance of acquired characteristic. In this one, instead of blending like a blender completely, right? You mix both. In this case, they're talking about how parents' physical characteristic is modified slightly and then passed on to their offspring. Now, after the chapter of meiosis, you should be able to tell me which one of these two hypotheses is correct. What are you guys saying? Why don't you vote one or two? There are three people. Come on, based on meiosis, we just learned which one of these hypotheses is correct. Very good, very good. Number two, right? Now, the modification of parental genes. Do you remember what process in meiosis allowed the modification of parental genes? Anyone remember the name of the process that allowed modification of parental genes? Right on, right on. Crossing over, right? Because of the synapses between the homologous chromosomes. Okay, so let's talk about what kind of genetic crosses there are. So for this class, all you need to know is two, and you focus on the first one, monohybrid cross. So mono means one, so they're keeping track of one characteristic. Dihybrid means two, so you're looking at two separate characteristics, okay? Now, Mendel, he figured out three principles of inheritance. They are dominance, segregation, and independent assortment. Now, I want to remind you at this time, Mendel doesn't know, doesn't know what DNA is. You guys know more than he does. He doesn't know cells. All he has is a bunch of pea plants that he grow and math. He can figure out how often he see a certain physical characteristic, what happened when he crossed one pea plants with the other one. But like I said, he doesn't know about DNA or cells. But but regardless, he was able to figure out these three principles that's related to meiosis. Let's talk about monohybrid crosses. So his first experiment involved crossing pure lines, means purebred. The other way to say it is pure lines, purebred is the same thing. So these are some terminology you need to know. P1 means the parental generation. F1 is the generation after P1. And then of course F2 is the one from F1. Okay, so you need to know all these terms. So what he did was he crossed round seeded plants with wrinkle seed plants. Okay, this is the P1, the parental cross. And then all the F1 offspring look very different than what it would be if it was blending inheritance. So let me show you what it looks like. No, that's what he did. He did a round seeded plants. He crossed a round seeded plants with a wrinkle seeded plants. But the first generation come out all look like the round seeded. The second generation, he got a 3-to-1, 3 rounds and 1 wrinkle. Okay. So remember the first slide I showed you about the 2 hypotheses? So what he was thinking of is this experiment right here already disproved the first hypothesis about the blending of the parental physical characteristic. If the first hypothesis was correct or were correct, what you will see in F1 will be a wrinkle seed, will be I should say will be a phenotype that looks between the two. So it's not exactly round, but it's not exactly wrinkled. Maybe it's a less wrinkled seed like this, but it's not. But F1 looks exactly like the male round seed, right? So then he figured, or maybe he figured that tells you the first hypothesis is incorrect. So the second one maybe is correct. But the question he has at this point is, why the wrinkle characteristic disappear in F1 generation and then reappear again in F2? Okay. And from this experiment, he actually figured out the law of dominance. Now, this is a picture showing you if the first hypothesis about the blending inheritance is correct, that's what the offspring should look like. It's not exactly perfectly round, but it's not very wrinkled either. It's a characteristic in between the two, right? But it's not. That's not what he got. Now, in genetics, you hear a lot about dominant, recessive, okay? So the definition for dominant is the characteristic that you can see that always show up. And the definition for recessive is the one that when it's paired with the dominant allele, it doesn't show up. Okay, so in genetics, when you're looking at a dominant characteristic, for example, black hair, black color hair. Okay, so the black color, since it's dominant, will use the uppercase B. Okay, big B. Let's say red hair is recessive. Okay, so to represent red hair, we don't use R. We use lowercase b. Okay, so the recessive gene we always represent with a single letter that is the lowercase of the dominant. Okay, so like I said, for red hair, it'll be 2 small b because it's recessive. Okay, just remember that because a lot of you will give me 2 different letters. So for each physical characteristic, you have a genotype. I'm going to talk about that later. That represent by 2 different alleles. Okay. Now allele, some of you came across this term in the PCR lab asking me what it is. It just said, like I said, it's a version of a gene. So let's say for eye color gene, you can have a version of brown color or a version of green color or a version of blue color. So it's just different variety of one gene, okay? So that's allele. Genotype is the genetic sequence of an individual, so it'll be unique, right? Phenotype is a physical characteristic of an individual, and that is because of a genotype, right? Genotype show up as a phenotype. Now, in genotype, when I ask in genetic questions in the exam, I ask what is the genotype for this individual, and it always have 2 alleles, because right, mom and dad, you have mom. Let me turn on the laser pointer. So it has 2 allele because you have 1 allele from your mother. Somatic cells, remember diploid, we keep talking about it, and now in genetic you can see it. You have diploid cells in somatic cells. So in somatic cells you always see diploid, and you have 2 alleles, 1 from your mother, 1 from your father. Okay, so when we talk about genotype, you always have 2 letters. Okay, and remember these 2 letters follow the rule of the law of dominance. The dominant allele, black color, brown color, whatever it is, or the round shape will be the uppercase letter, and the recessive will just use the lowercase of the dominant allele. Okay. And if I ask for phenotype, it's always a descriptive word: round, tall, yellow, red. So it's never two letters. So just remember that. Try this question. Good, everyone. Is B, right? I keep talking about each physical characteristic is controlled by two alleles. Two alleles. Now let's look at the word homozygous. Homo means same. Homozygous means that for a particular allele that you're looking at, you have the same allele here. This is an example of homozygous. So both of these letters will be identical. So it could be two big letters or two small letters, right? And these are called purebred or pure line because there's only one kind of allele in this genotype, okay? Versus what we call heterozygous. Hetero means different, right? So if you look at this, heterozygous will have different alleles for a particular gene. For example, you have a big H and a small h. Okay, that's heterozygous. Now let's look at law of dominance. Remember the experiment that Mendel did? He crossed a round seed parent with a wrinkle seed parent. So wrinkle seed he figured out is recessive. So that's why it's too small r. Okay, not W. Remember that? So since round is dominant, that's why it's big R, too big R. So they are both homozygous. So homozygous could be homozygous dominant like this. Both of these letters are big or uppercase, or homozygous recessive. Both of these letters lowercase. Okay. So when you cross these two parents, what you get is a heterozygous F1 individual. And since the law of dominance works, it tells you in this case, the small r, the wrinkle, will not show up because it's recessive. So only the round shape will show up. That's why all the F1 seed looks round because it's a heterozygous dominant. Can you see that? This I already talked about. So you can be homozygous dominant or homozygous recessive. Okay, and this is how heterozygote is the same thing. Oh, so this picture, let's see if there's more thing. Okay, this picture is talking about to look like a dominant, I should say, to have the dominant phenotype. You can be homozygous dominant, or you can be heterozygous dominant, because this small t is not going to show, you're only going to show the big T in phenotype. Okay, so if you're this person, it is heterozygous dominant. It's called heterozygote. That's what it's trying to tell you. So these two both can give you the same phenotype, even though the genotype is different because of the law of dominance. Give me a minute, I need to fix my internet. Somehow my internet wants to switch. Okay, hopefully now it's good. Let's try this question. Let's see. All of you say D. Good. Okay, you should know that, yeah, D is correct. So one of the phenotype, the round seed, is always dominant over the other one. What about this one? So for a true breeding, which is same as purebred, pure line, correct, the answer is C. So for any kind of purebred or true breeding or pure line, it's either homozygous dominant, which is two dominant allele, or homozygous recessive, which is two recessive allele, okay? Is C. So this question is to make sure you guys understand homozygous is not just for dominant. It can be homozygous recessive too. Now we have this question. Why don't we all answer number one first? Oh, let's do number one and number two. Do two at a time. So you guys type your answer for number one and number two together. Yes, so the first one is 2i is dominant because all the kids have two eyes, right? And this is F1. So let's do the next two questions. If you use the letter E for this gene, what's the genotype of the parents first? And then what's the genotype for the offspring? Okay, let's do number 3. What is the genotype for parents and then the offspring? We're working on number 3. What is the genotype for the parents and then the offspring? Yeah, so far everybody got it right. So the one eye is E, one eye parent is two small e, two eye parent is two big E, and the offspring is a heterozygous, one big E, one small e. Good job. So what about number 4 now? If you cross these offspring with each other, what percent of the new offspring will you see to have two eyes? We're looking at percent. Yep, 75% is correct. Now, so we just did principle of dominance. So from looking at the pea plants that Mendel crosses, Mendel was able to figure out that there are two alleles. Number one, so one allele is dominant over the other allele because he figured out if you only see round in F1 generation, that means the round allele is dominant over the wrinkle allele. Now remember, he did all this just by looking at math and his logic. He doesn't know anything about DNA or cells, right? Um, so to explain, remember how when you cross heterozygotes, like the one we just did with the two-eye babies, they all have this 3-to-1 phenotypic ratio, so 75% to 25% in the F2 generation. And from looking at this math, Mendel was able to reason that each one individual has two alleles, and not just that, he already figured that one out in the law of dominance. He figured out that these two alleles actually separate from each other during gametes formation. So he's talking about meiosis right here. He's talking about how the homologous chromosome separate into two different gametes. Okay, so that's the law of segregation. Now, just to be sure that you get it. So remember in the homologous chromosome, we have a paternal, blue always is father, red is always mother, right? And these two are homologous because they're the same size, same centromere location, which is right here. Good. And then they have the same locus of genes. That means the gene location will be the same. If this is eye color, they both have eye color gene at this exact spot. But like I said, they can have different alleles. One could be blue, the other one could be brown, right? And during meiosis, these chromosomes is going to make a replicate of itself. Let me turn on my pen. So what will happen is at centromere, you have another one here, replicated chromosome like this after S phase, right? For mother, and then for father, I can actually pick color. For father, the sister chromatase will be attaching to the father at the centromere like this. So during prophase, you have these tetra. Remember, you have these two replicated homologous chromosomes forming tetra synapses. So they can exchange, the blue and the red can exchange genetic information. So the mother is a little bit of father, contain a little bit of father material, and the father contain a little bit of the mother material. Okay, and this is another picture showing you. Same picture here. You have one type of chromosome here, okay? You have the mother and the father. After S phase, they replicated, right? So then they form tetra, go into metaphase 1, and then the homologs get separated into two separate cells. Now remember, this at this cell here is 2N, because mom and dad is still together. This cell here is also 2N, a diploid, because mom and dad is together again. Now, when you go to here, you become haploid because mom and dad is separated. For each type of chromosome, you only have either the father's copy or the mother's copy. They're never together. Now, if you look at this picture closely, we're looking at allele, right? So in the father, you have a dominant allele, big R. Mother, you have a small r, which is the recessive allele. So at the end of meiosis 1, you see how the dominant allele is separated from the recessive allele. So when you make gametes at the end after meiosis 2, all the dominant allele will be in one gametes, will be in these two gametes, and all the recessive allele will be in these two gametes. So now you understand when we keep talking about in meiosis, how the homologous segregate is very significant because it turned out homologous chromosome can carry different alleles, even though they're very similar, but they don't carry the same genes, right? So that's how different genes get segregated into different sex cells. So when then the sex cells go combine with another sex cells, that's why you have all these different combination of alleles. So this is just showing you at fertilization, I keep telling you that when the different alleles combine, right, they restore the 2N. Now this is diploid. This is haploid, right? Two haploid sex cells combine now become a diploid. Now you have combination of alleles from two different parents. Segregation. Good, it's C. Okay, let's do this concept map. How should we do it? So how many people do we have? I think we have, yeah, so C is correct. We have 9. Do we have 9? 1, 2, 3, 4, 2, 3, 4, 5, 6, 7. I think we have 8 or 9 people. So let's do this. Okay, Misty, can you do this circle here? Physical form. Okay. Heath, can you do this one? Genes for a trait here. Heath will do this one. Segregation of alleles. Dahlia, you can do this one. One copy of each gene. Who do I have? And Neri, you can do this one. Saul, you can do 2 copies of each gene. In, can you do express in heterozygote? Sam, can you do this one? Form of a gene. Who else do I have? Okay, Harley, can you do this one here? Hidden in heterozygote. And then that's all the people that I can see. Okay, let's look at what you guys have. Physical form. Physical form is supposed to be what? Phenotype, right? Physical characteristic, this is supposed to be a phenotype. Genes for a trait, you got it right, which is genotype. Segregation of allele, Dahlia got it right, is J. Mendel's law of segregation, 1 copy of each gene is haploid. 2 copies of each gene is diploid, right? Homologous together right here. Form of a gene, a version of a gene is allele. This one is allele. If those 2 alleles are same, they're homozygous. If those 2 alleles are different, they're heterozygous. And then you have expressed in heterozygous, which will be what? Dominant. Dominant allele will be expressed in heterozygous, and recessive allele will be hidden in heterozygous. Good job, good job. Let's go to the next one. Now, a lot of you already know how to do Putnam square. Putnam square is to help you figure out the genotype and phenotype of a monohybrid cross or dihybrid cross. But let's talk about a Putnam square in details. because you may be doing it without knowing why you're doing certain things. Okay. Now, number one, you will put one parent's gametes on either this side or this side. Okay. So if this parent's, let's say parent one, he is a heterozygote. So that's what his genotype look like in his somatic germ cells. Well, I should say in his germ cells, right? The 2N diploid that start with. So when this parent's germ cell with this genotype make gametes, let's say sperm here, those two alleles is going to segregate because those two alleles is going to be in they are on homologous chromosome. So the big Y will go to one gamete, just like the one I showed you, because the law of segregation, right? So the big Y will go to one gamete, the small y will go to the other gamete. Now parent 2 showing you the same thing here. This parent, the mom, could be a heterozygous. So same thing, law of segregation will separate the big allele and the small allele in two different eggs. So the top and the side here is showing you what the gametes will look like genotype wise in each of these parents. That's why you write individual letters like this. Okay. Now when this sperm and this egg combine to make a baby, you restore the 2N. This is haploid 1N, you restore into 2N. You combine the two. Okay. So you can see that this baby will be 2 big Y. So that'd be a homozygous dominant, right? And you do the same for the other square. So you can see how you go from N here, when they combine the two, become 2N right here. Now, based on the law of dominance, you can figure out what the based on the genotype, you can know what the phenotype will look like. If Y is dominant, which is yellow, and then this guy is homozygous dominant, has to be yellow. This one is heterozygous dominant, so it has to be yellow too. So you already figure out based on the genotype, these three babies will be yellow. This one will be green. Okay, so with Putnam square, you're using the law of segregation and the law of dominance at the same time. Get it? Now this is what I just talked about. Each parent, now remember this is showing you the 2N germ cell. What you start with is a diploid. When they go through, after they've gone through meiosis, they'll have haploid, sperm or egg, right? And then these two alleles will segregate because they're homologous chromosomes. Now we're going to talk about later on what if the alleles are not on the homologous chromosome, and then you will have the dihybrid cross. But this is exactly what I just mentioned. You have the eggs and then the sperm. Now this is what Mendel did. He did, and I know you guys already know the answer, but on paper, that's what it looked like. He has a homozygous dominant and homozygous recessive as parental P1 cross, right? So if you segregate these two alleles into here and those two into here, you end up, that's why you end up with all heterozygous dominant round seed, right? That's why everybody look like round in the F1 generation. Now then when he did the second round of crossing, remember the wrinkle disappeared and then it reappeared again in F2 when he crossed the F1 generation together, right? So that F1 now is heterozygous. So when you do the law of segregation, half 50% of the gametes will be big R, 50% of the other 50% will be small r. So then when you do the cross, right, that's how you get a 3-to-1. So these three phenotypically will look identical, even though genotypically they are not the same. Can you see that? This is homozygous dominant. Heterozygous dominant will look identical to homozygous dominant. So three of these guys to one of the homozygous recessive. So the wrinkle seed reappear in F2 because some of the F1, the recessive, get to combine in this case, and then that's why the wrinkle appear again. Otherwise, it's disappear in F1. So Mendel was going, what in the world, where did it go? It's hidden until, so you don't see recessive showing up in a phenotype situation unless it's homozygous recessive. Because if there's any big R, it's never going to show up. So two small r has to come together for it to show as a wrinkle. And that the chance of having this is very, very small. That's why it's 3-1. The 1 is the two small r's together. Now you guys do this question. See if you get it. Look at these two dogs, really cute, and it tells you what the genotype is for each one. So when I'm asking which allele, you should answer either the big H or the small h, right? Because in this case, you don't really know which one is dominant. I mean, as far as I didn't say, what it stand for. Anyone else? 50-50 chance, either the big H or the small h, right? Yep, so I see a lot of you say big H is the answer. And you know why, right? Let me see, I switch it to a pen or something. Now, if you read the question carefully, it says that when you have two, let me do pen. When you have two Chinese crested dog, so it's this guy, right? So you're crossing heterozygous big H, little h together. They're dead puppies when you do that. Now, if it's showing you two small h give you puppies with hair like this, and then heterozygous give you puppies with this. So obviously, if you have two, I don't know why it looks like this. If you have two big H together, those are the only other one that it could be there, right? That then they have to be dead. So big H is the one when you have them together. It will kill the puppy. Discard. So good job. Big H is correct. So let's try this question. Now remember, Chinese crested dog is the one on the right without hair. And just in case you forgot, it's the heterozygous one. So the breeder wants the most hairless dog with the least dead puppy. How would you, what kind of cross would you do? The key word here is, what do you think the key word is? They don't want any dead puppies. Live puppies can be sold, but that one you can't do anything about, right? The key words least dead puppy and, oh, I still have it. Most Chinese crested hairless. So he wants the most number of hairless puppy and least that one. Yep, C is correct. Now, A is not correct because that's what we just did before. If you have two heterozygous, you always get, if you cross two heterozygous hairless dog, you always end up with one dead one, right? But if you cross two powder puff, which is two homozygous recessive, you don't get any hairless dog. So the only way to go is C, to make a hairless with a powder puff. That way you avoid all the big H. Good job. Now, when we talk about independent assortment, we're actually looking at, if you remember in meiosis, when does independent assortment occur? Do you remember? During which phase in meiosis does independent assortment occur? Yep. Right on. Thank you. So it's metaphase 1, when the tetra line up randomly, right? So when we talk about independent assortment, we are talking about uh, two, two tricks that is on different chromosomes. So far we've been talking about alleles. Remember, alleles are the different version of the same gene, right? So, but what if those genes that you're interested in is on two different chromosomes? Let's say one is on chromosome number 1, one is on chromosome number 20. So the principle of independent assortment is telling you that the way that chromosome number 1 assort into different gametes is completely independent to chromosome number 20. So let's see if I have a good picture. I think you guys did this question already, right? Actually, this question is incorrect, so ignore it. So let's look at independent assortment. So we're looking at two different characteristics that's located on two different genes, on two different chromosomes, okay? So here, so that's why we're looking at dihybrid cross here. Dihybrid cross here. So in this example here, we're looking at the characteristic of the shape of the seed and the color of the seed. See, one could be round or wrinkled. That's the shape of the seed. And the color of the seed can be either yellow or green. Okay. So, and these two different characteristics is on two different chromosomes. So on one of the chromosomes, for this male parent, it could be homozygous dominant, and on the other characteristic is homozygous. So for each characteristic, just remember in dihybrid cross, for each of the characteristic, you have two alleles for each in the germ cells of the parents. Okay, same thing, right? This is looking at homozygous recessive. So for each characteristic, you have two alleles because this is still 2N diploid cells. And when they make babies, when they make gametes, you can see that how these two, the two small r segregated into a small r and then a big R. Let me show you another picture. This is a better picture. So you're crossing a heterozygous, big R, small r. So that's for one characteristic. So for the seed shape, each parent is heterozygous dominant. For the seed color, and each parent is heterozygous dominant again. So what in the hybrid cross is telling you, when these germ cells, diploid germ cells, form gametes like this, you have four different combinations of gametes. So what they're trying to tell you is this big R and little r is going to segregate into gametes, right? They have to separate. Big Y, little y has to separate. But when they separate, big R, let me change pens. Big R can go into the same gamete with the big Y because they're on different chromosome. So there's no they randomly assort. So the big R and big Y randomly assort into different gametes. So that's why you have these four different possibilities. So big R can be assorted into the same gamete as big Y, or big R can be assorted into the same gamete with the small y. So you have, that's why you have this big R, big Y, big R, small y. Now remember, these are possibility. It's kind of like gambling. And the same thing, the small r can be segregated together with the big Y, or it can go with the small y. We don't know that. Depends on how they orient. So that's why you get these two other possibility of gametes. Okay. Now for genetics, for this class, we're not going up to more than two characteristics. So we're not going to go over and above dihybrid cross. We're going to stop right here because you can see that with just one set of allele, you get 4 squares, right? But with two sets of allele like this, you're already up to 16 squares. So the top is just telling you the possibility of gametes from each one of these parents. And the key thing to remember is this. Each one of these allele from the pair has to combine with the other one pair like this. So you get one of each. But the thing is, you don't know which one you're going to get. That's why I'm going to lay out all the possibility here. So same thing happened here. And when you combine, you can see that you have 16 possibility. And the ratio in a heterozygous cross is always 9, 3, 3, 1. Okay, so you can see all these. So this is wrinkle. This is showing you wrinkle and green, right? And all these is just wrinkle and yellow instead of that. So this is just like what I talked about, the two the genes that they're looking at, the two genes that they're looking at are sort independently of each other during metaphase 1. Now I'm gonna have you guys take a break for 5 minutes, and I'm gonna come back and talk more about dihybrid cross. Okay. 5 minutes break. Okay, let's get back together. So here's a picture to summarize what we just talked about, the principle of independent assortment. So here's the first picture in metaphase one. Let's see, where is it? In metaphase one, now these supposed to be in tetra, but it's not showing you as tetra. So this, I think these two are metaphase one right here. Okay, so in metaphase one, remember these tetra, which is these two homologous chromosomes, same size, right? These two homologous chromosomes. And can you see that? Can you see that what they're trying to say is the round and wrinkle characteristic is on this big one here. Okay, the black dots here on the green and the dark blue one on the blue one here. And then the seed color, which is yellow or green, is on the smaller chromosome on the bottom. So let's call these first set here is chromosome number 1 because it's bigger. And let's call this set here, let's say it's chromosome number 10. So what they're trying to show you is these two genes, one is the seed shape, one is the seed color, is on two different chromosomes. So that's the first thing they want to show you, okay? And number two things they want to show you is that the homologous chromosome, the blue and the green, carry different alleles of this gene. So the blue one carry the dominant allele, the green one carry the recessive allele. So this guy is round, this guy is wrinkled. Okay, same thing on the bottom. The blue one here carry the dominant allele for the seed color yellow, and this green one carry green. Okay, it's recessive. Now, so do you remember in the independent assortment stage during metaphase 1, the way that get my the way that the tetra orient to the poles is random. So in this scenario here, you see that all the blue, all the paternal is facing this pole here. Okay, so when it separate during meiosis 1, you get all the blue, the paternal alleles in one cell, all the maternal in the other cell. But the other possibility is that you could have them facing the pole crisscross like this, right? So when you have formed gametes during meiosis 1, you have half of the paternal and half of the maternal. So if you follow the alleles along, you will see that in this scenario, this gamete will have all big letters, big R and big Y, right? One of each. Remember that when at the end, there's 4 more here. This one split into 2. This one split into 2. So for this one, you get 2 gametes that both have big R and big Y. For this one, you have gametes both have small r and small y. Can you see that? Now, for this one, because you have crisscross, so when they form 2 gametes at the end of meiosis 2, you have 1 big R and 1 big Y. And this is 1 small r and 1 small y. And that is exactly what I show you here. You can have all the big together, big R, big Y, or 1 big, 1 small, 1 big, 1 small, 2 small. Okay, this is what this picture is trying to show you. So you just remember for each gene, one allele always go to one of the gametes, okay? But since human, you can imagine we have 23 pairs like this line up. So the amount of maternal and paternal alleles in each gamete is going to be very, very different. It'll be very different. And last time I showed you the math, we have 8.4 million different combination of gametes because of this. Okay. Now, before I go on, is there any question about independent assortment? I think this one is kind of harder to understand. Now, we're not going to do this kind of question. I think it's not very beneficial. So let's skip this. Okay, here's the summary. What we learned this morning, we learned the law of dominance that help you to predict based on a genotype what a phenotype will look like, right? And then we also learned of the law of segregation and that the law of segregation will give you the ratio of 3 to 1, 3 dominant to 1 recessive, and that also explains why the wrinkle seed disappear in F1 generation. Now we just learned the law of independent assortment, and this one, remember, only apply to dihybrid cross when you're looking at two different characteristic, and that is located on two different chromosomes. So the way one chromosome will segregate uh, is completely random compared to the other one. So there's no a fixed way that big R always go with big G or small g, just random. It can go with big G or small g in this case, okay? So in this case, you're 16 squared and always give you a 9, 3, 3, 1. So the recessive now is 1 of 16, completely recessive. And all the rest, these two in the middle, is a mix of both, right? And this one is completely dominant, 9 out of 16. You get round, which is dominant. Wait, where is it? Round, dominant, yeah. Yeah, so you get you get 3 out of 16, sorry, 3 out of 16, that's completely dominant. You have brown and yellow together, okay? So I'm gonna wait for you guys to come up with a question. If not, I want to look at the worksheet first, and then we're gonna talk about the lab. Brown, brown rabbit. Yeah, brown, going. Okay, so it doesn't look like you guys have questions, but so let's look at the worksheet first. Now I'm gonna help you do the last question, but let's look at the first few here. Make sure you guys are understanding what I'm getting at. Um, for question number 3, a lot of you get it wrong. Okay. Remember, in this case, for example, I say that what? I ask you to list the genotypes, and straight hair is dominant to curly hair. Okay. So how would you write the genotype here? Yeah, what? Give me the answer for this one. What would the answer for this one? So let's make sure you get it right. Well, what letter will you use? And Neri use R, but why R? Where does it come from? Usually people just use the letter for the descriptive word. So in this case, so do I have more? So Heath is good. He said big two big S, but here's the problem, okay? So that's why I said most of you get this one wrong. If straight hair is dominant to curly, what kind of genotypes can you have to get straight hair? You get, so you guys get the first one right, which is two big S, right? But there's another one that you can have. The letters vestiges come from, let's say this is straight, right? Straight hair, you just take the S from straight. That's it. If curly is dominant, you just take the C from curly. So it's basically from the adjective, the first letter of the adjective. But you can make up any letter you want as long as you indicate. For example, if Anari say she wants to use R, then she have to say R equal straight hair for me. Okay, so what is another genotype that you can have to have straight hair? Remember, you can have homozygous dominant, and what other genotype you can have to have straight hair? Come on. Yes, so for this one, you can have two big S, one big S, and one small s. So you can be homozygous dominant or heterozygous dominant to have straight hair, right? Now, so if it's recessive, like in here, in this case, it's curly. You can only have one genotype to have curly hair, and what would that be? There's only one genotype for recessive. Good. So it's two S, small s. Remember, Misty? I said in the beginning that you want to use the dominant letter, but you just change it to lowercase. You don't. So in this case, you don't have two C. Just like in this case here, you won't have a big S and a small c, right? Because you just use the dominant letter, and then lowercase will represent curly for that case. Okay, remember that. You only get one kind. 2S. Good. So your no 2C. So I missed you. Remember that. You use the dominant letter, except you use the lowercase of that letter. Good. So same thing here, okay? Remember, so you guys always miss this. It's this part here. That when you think about dominant, you always give me the homozygous dominant. But remember, for dominant phenotype, you can either be homozygous or heterozygous. So we're good with this one. That's why I want to point it out. Now, the other one that you guys get it wrong is the number 4 here. Okay, number 4 here, I'm not asking to do a Putnam square, and all of you, for some reason, will set up a Putnam square for me. Because if you look at this here, Putnam square setup is starting in question 5. Okay, so in here, number 4, all you have to do is to show me the genotype, let's say 2 big F, 2 small f, whatever it is, equal what kind of phenotype. Okay, so let's do one for you. So the question is the allele for fire breathing is dominant. So I will use F for fire breathing. So big F will be dominant. Small f will be recessive. Okay, so dragon can be fire breather or non-fire breather. So I will have for my first one here. So I can have 2 big F, correct? And that will equal to something. I can have a big F and a small f. Remember the small f represent the recessive. I can have 2 small f. Oh, 2 small f. So these are the 3 possible genotypes for a dragon. Now, there's only 2 possible phenotypes for the dragon, either breathe fire or not breathe fire. So let's try 2 big F. Is it going to be fire breather or not? You should get it, right? Are you understanding what I'm trying to get at in number 4? So that way you set up all your genotype and phenotype in number 4, then you can go on and do number 5. So 2 big F, since fire breathing is dominant, so 2 big F has to be fire breather, right? And so would this one. These two be the same. This will be heterozygous dominant, homozygous dominant. Now 2 small f is homozygous recessive, so this is the only one that is non-fire breather. Getting it? That's what I want. Yep. So that's what I want in number 4, so you guys get it. So I'm going to give you a minute to make sure you get this, and I'm going to go on the last question on this worksheet. Okay, let's go look at the last question, and the rest you should be able to do, but the last one is a dihybrid cross. So I want to make sure that you guys get it. So if you look at the dihybrid cross, this is the question it's asking. Right, you have a bunch of rabbits, and they tell you all the genotype for each particular phenotype. Right, so the question is talking about if let me highlight it, if a deformed brown rabbit, so for deformed, it tells you here is big C, little c, right here. Brown is, look at this, is big B, little b. So one of my parent will look like this as far as genotype is concerned. Now these 2 characteristics is on different chromosomes, right? And then it's going to cross mate with a deformed leg again. So I have big C, little c, but this time it's a white rabbit. White rabbit is what? Here, 2 small b. So that's what I have. So this parent's genotype will look like this. Okay, now, so when you do your square, not laser point diamond pen. When you do your square, since it's a dihybrid cross, you can automatically draw a square. Oh, dang it. With 16 smaller squares. 2, 3, 4. 1, 2, 3. Okay. And let's do this parent on this side here. So the thing is, this allele can segregate into the same gamete with this allele or this allele. So in this case, I'll have a big C and a big B, right? Because remember, each gamete only get one of the two allele in each characteristic. So then, or this big C with this small b. So that's the first two possibility. Now, when you look at the small c allele can go with the big B, or it can go with the small b. So in this case, I have the small c with the big B, and the small c with the small b. Can you see how I do all the gametes? So there are 4 possible gametes in this scenario with 2 different characteristics and with these alleles here. So you will do the same thing for this parent here on the top here. Okay, remember the big C can go with either these two small b, but they will look the same. Small c can go with either one of these small b. And you write on top like this here, just like what I did. And then you combine the gametes inside the square, and then based on this here, you can figure out, so it's asking about the probability of D form. So you're looking for bunny, that's big C and little c, and brown. So you're looking for a bunny, that is big B, little b, right? So that's the first question. What is the percentage of bunnies will be having these two genotypes or phenotypes here? Now, also asking you about how many will be dead out of the 16. So you need to tell me both of those. Is there any question about this question? Now, if there's no question, I want to go to the lab. So today we do lab 12, which is genetics. And for today, I think we're only going to do Yep, Heath, right on. If you read the instruction, it tells you which one is that. I think we do part 1. Let's see, part 1. We do part 1. Okay, for today, you just need to do part 1. And let me show you, I think you should have the PowerPoint for genetics already. See where it is. Lab Wait, do I have the lab? I think the lab is very easy. Let's do it. I think I have the lab here. Where's my lab? Okay, let's look at the lab. So today you're supposed to do part 1. So let's look at part 1. It's pretty straightforward. So at home, you guys can use a penny and do this first part, right? You toss a penny into the air 10 times. And then you just write down how many heads and how many tails you get. Now, so the second question is asking, does the first toss have any influence on the second one? And you guys can answer all these questions. Now then you can do part 2 by tossing 2 pennies at the same time, right? And what they want you to do is, now why do they use a penny? I think it's kind of silly. So a penny has a head and a tail. So that tells you if penny were an individual, it will have a heterozygous genotype, a head and tail, H and T, or big H and small h. If head, you would say is dominant. I don't know. If you say tail is dominant, then you have big T, small t, whatever you want to do. But say that's the first. So the second one is just 2-person, or well, I guess a penny. Here they represent a particular gene. So for each gene, so I shouldn't say a person. It's talking about a particular gene. So for each gene, you can have 2 alleles, right? You can have head or tail. So that's what they're trying to get at. You toss 2 pennies. Each individual or each gene has 2 possible alleles, head and tail. So in this Putnam square here, you just put an H and a T, and an H and a T, and then you can figure out what the offspring is like. Now this one, instead of toss the 2 pennies 20 times. So you just follow what they ask you to do. So we are not going to do the class data because we don't have a class. So you just toss your 20 times and just write down your number and your frequency. And we don't have to do B yet. Question number 3B. We haven't talked about codominance and incomplete dominance. We'll talk about that next time. So this lab, we'll finish it next time. But today you just go all the way to, we're not doing question 4 either because we don't have class data. So wait on 3B and cross out question 4. And in this one, question 5 is easy. Okay, so remember for each female, you have each female, the eggs will have one of the X chromosome because you have the X. In diploid, you have two X together, right? So in haploid, you have one X here, one X here. And in male, it's same thing. In diploid, you have X and Y here, but then in the haploid state, you have either X or Y. So if you do that, you figure out the probability of having a boy. You already know that it's 50%, but now you can make it legit by showing it through the Putnam square. Okay, so you do that and you figure out that it looked like exactly 50%. Okay, now here number 6 is talking about um, rule of multiplication. So this is very straightforward. What it's saying is that the let's say you have the chance of having a boy is 50%, right? Or a girl is 50%. But if you want a particular event to happen a certain way, so let's say in this case, in number 6, is that if you want to have 4 boys in a row, so the chance of having the first boy is 1/2. The chance of having the second boy is 1/2. But you have to multiply all these probability together, not add, because it's harder to get 4 boys in a row, right? So that's why it's 1/16. So the number gets smaller and smaller if you keep multiplying. So that's what the rule of multiplication is. It's telling you that if you want a certain event to happen in a particular order, like this, 4 boys in a row, or you toss your penny 4 heads in a row or 4 tails in a row, it's going to be harder than just head or tail. Okay. So in this question number 6, what about 5 sons in a row? How will you do it? So you just do the same thing like here on top. You have 1/2 times 1/2, but this time you have 5 1/2 multiplied together. And then you have a very small number. Okay, so we have 8 minutes left. So why don't you look over it, see if you have a question. I'll stay until 11:20. Remember, we're not doing photosynthesis, so we have quite a bit of time at the end for you guys to catch up and do stuff that you need to do to prepare for the final exams. And I hope that I know it's tough doing online, but with that extra time, I hope you guys will use it wisely to do better on the exam and the final exams. Okay, so if you ready to ask me questions. If you don't have any questions and you know what you're doing, you're free to go. If you have questions about the worksheet, about the lab, we can do it. We are doing part 2 but next time we'll do part 2. Because this is human genetics. We will talk about human genetics next time. Today we only talk about peas and and flower color and stuff like this. Next time will be for real. Canary number 7 of what worksheet? Let me see number 7. right? Okay, so it's on the worksheet number 7. So let's look at what they have here. So the parents both have wings, but the offspring doesn't have wings. Okay, so think about this. If wings are dominant, right? So for two dominant or two winged dragon to have a wingless dragon, the parents have to carry the wingless gene or alleles, right, to give it to the offspring. Does that make sense? So if you look at this, in order to get wingless kids from winged dragon, the parents cannot be homozygous to big W. Because if that's the case, where is the small w come from for the kids? The small w for the kids has to come from the parents somehow. So the only logic in here is you have big W, small w for each of the parents. So you have big W, small w crossing with big W, small w. So both parents has to be heterozygous dominant. So then they can give the small w from each parent to the offspring to become 2 small w. You see that? 1 small w from each parent combined become 2 small ones. So this is the 3-to-1 ratio thing. And you say that the both parents cannot be small w. If both parents is small w, they cannot be wing. Wing is dominant, so they have to be either heterozygous dominant or homozygous dominant. Just remember that because it tells you here, wings are dominant. Oh, yeah. So no, when they ask questions like this, they're just telling you one of the offspring. It's never all the offspring. See, produce an offspring. Any other question? Oh, sorry, and the study guide is not updated quite. Let me look at the PowerPoint. Where does it say autosomal phenotype? Is this the study guide you talk about? This PowerPoint study guide didn't say anything about X-linked phenotype. Did I give you the wrong PowerPoint? Well, but anyway, the autosomal phenotype and X-linked, that's what we're going to talk about next time. Oh, okay. So the lab PowerPoint, that lab, remember next time in part 2, we're going to talk about autosomal uh, phenotypes and X-linked. So next time, part 2 of the lab, we'll do human genetics. So that's where the X-linked and the autosomal phenotypes comes in. So we'll talk about that next time. Misty, it depends on what the question is asking you, okay? So if the question only asks you about wing, then you just do a Putnam square for wings. If the question only asks you for fire breathing, then you only do a Putnam square for fire breathing. I don't think in the dragon questions here they ever ask you for dihybrid. If they ask you for both, you do both. Just like in the question that I did with you guys with the rabbits, the deformed leg and the coat color. But in these dragons questions, it never asks you to do both. Okay, last chance. Any more questions? If not, I'll end the live stream. And I'll see you guys again on Friday. We'll talk about human genetics. So I think that's way more interesting. But you have to get through today's material to understand Friday's material. And we'll finish lab 12 on Friday too. Yeah, we're all good. Okay, I'm gonna log off then. Okay, Neri, what do you have? Let's see. On number 1, are you talking about the lab? And Neri, are you talking about lab number 1, not worksheet number 1, right? I think it's lab. Let's see, lab number 1. What question do you have for lab number 1? You just take a penny out and toss it 10 times. And then write down how many times you get a head and how many times you get a tail. Part B to D. So this is exactly what they're asking you. When you toss the penny the second time, does the first toss have any influence on the second? There's none, right? Doesn't matter what you get on the first one, it won't affect how the second one turn out. So it doesn't affect. That's what is a very straightforward question. Yeah, so you got to raise no influence. Does the third have any? Well, none of them has any influence, right? Every toss is an individual event. You either get head or tail, and whether you get a head the first time doesn't influence the second or third or fourth, whatever it is. So D here saying that if you toss the penny 10 times, out of the 10 times, how many times do you get a head? Let's say you get 5 out of 10. Then this will be 50%, right? So 5 over 10 will equal 50%. That's what they're asking you. So if frequency of tail you get 3 times out of 10, then it'll be 30%. Okay, I'm gonna end the live stream now. You can always text me questions or email me questions.